Hey guys, so we're going to continue with our DNA barcoding process with part 3, which is electrophoresis. So the materials that you're going to need for this are the PCR tubes that we did in the last step. We're going to need this e-gel power base, which is plugged in to our outlet here with this power cord. You are going to need a molecular weight marker, this purple thing, and you can put it in another tube later if you need to. You'll need your micro pipettes and their tips right here, and we'll have a waste bucket also to put the tips in need of disposal. We'll have distilled water here and our microfuge for our pulses that we're going to need to do. And the last thing you're going to need is just a permanent marker to label your tubes as necessary. Alright, so we are on step A as of right now. Refer to your instructions for the logistics on this, but basically what I did was for our sample tubes, I added 5 microliters of our PCR ones that we spun earlier, and then 15 microliters of distilled water. So the final volume in each of these should be 20 microliters. And for the first part of step B, in this clean tube here, I put 10 microliters of the marker and then 10 microliters of distilled water. So the final volume on this one should also be 20 microliters. Okay. So we are going to continue with plugging our little e-gel thing here into the power base. So what we're going to do is we're going to remove it from the package, and then you're going to take this little comb here off the thing. One important thing to remember is to make sure that you don't take off this comb until all student samples are ready. So what we're going to do now is using the right side first. Has little places to plug in, kind of like a battery. So then you're going to click that in, make sure it's even on both sides, and a red light should appear just like this one when it's all ready. So the next thing what we're going to do is we're going to load all our 20 microliters of samples into our e-gel here. So one thing you can notice is it's okay to write on this. You can, in fact, it's better that you label which samples are going into which well. So we have the molecular weight marker in all 3 of our little samples here, and it's better to put it towards the middle of the e-gel. So what we're going to do now is my pipette is set to 20 microliters. Pick up my sample here. Collects every little last drop of the sample. This is sample 1, so I'm going to take it and put it in this well here. I'm going to do the same thing with all of my samples here. That all your samples are loaded, we're going to press the 30-minute button here, and this will start electrophoresis. And as you can see, the light goes on. It also might be helpful to set up an extra timer outside as well, so in case this isn't accurate. So after 30 minutes, this current should shut off automatically, and after that you can remove the gel cassette here to analyze the results. And that's your electrophoresis.
After your gel has finished running, you will disconnect it from power, pulling out that little plug, and then disassemble the gel by pushing it up on the left side and releasing. So now here's your gel. You can place the gel right onto the safe imager made by Invitrogen, and then make sure to cover up the light box imager, because the light is a high energy blue light, which is not good for your eyes. Then go ahead and turn the light out to view your results. Before we look at the gel under the blue light, I'd like to quickly talk about what you will see. First of all, we used markers provided by the kit, and these markers were made by taking a plasmid called PBr322 and digesting it with an enzyme, BSTN1. And with that digestion, you can see that multiple DNA fragments are generated. The largest one is over 1,800 base pairs, and then there's a doublet, 1,900, 2 of them base pairs, and then below that a 383 base pair fragment. The CO1 gene runs somewhere between 650 and 800 base pairs depending on the organism. So you would expect to see this fragment lining up just below. If you drew a line, virtual line, it would run just below the 929 base pair marker. The RBCL genes, which are derived using plant primers with plant material, are a little bit smaller. So if you see fragments that or bands that run well below the 383 base pair, these fragments, they're not DNA fragments. They're most likely leftover primer that has not been incorporated into a new fragment of DNA. The ones that appear up here, you can get excited about. So the last thing you'll need to do is to take a photograph of your gel using a smartphone with your flash off, looking through the amber light screen. Take a picture and make sure to include the origin or the wells at the top. And with that photograph, you can go ahead and analyze the molecular weight of each band using Logger Pro. And you will also need a photograph to send along with your samples to the sequencing company, GeneWiz.